This is a super, super interesting company. And we have everybody take your seats so I don't have to shut the door. Maybe shut the door. That's good. So many of you met Manuel Ebner last night. Remember Manuel? Very interesting. He's one of the co-founders, along with Dr. Vladimir Cmiljanovic.
Something like that.
And Andreas Eminger. Swiss Rockets is a fascinating biotech platform. We were talking earlier about, unfortunately, we never like it when we hear that companies have gone bankrupt, but we heard that 23andMe has gone bankrupt. Well, here we go. Swiss Rockets had nothing to do with it, by the way. However, Swiss Rockets is probably next generation. A fascinating company. There's a recurring theme here about how data is transforming each ecosystem and platform. Would you please welcome Vladimir?
Thank you. Thank you so much, Julie. Do you hear me well? Yeah? Excellent. So, thank you so much for our invitation. Hello, everybody. My name is Vladimir Cmiljanovic. I'm originally from Serbia, where I grew up in a family of entrepreneurs and team handball players. We all were professional team handball players. I became national team player as I was 15. With 19, I moved to Germany to Bundesliga. With 20 to Switzerland. Transferred actually both by cigar company Davidoff, and Schneider, the former owner who died on cancer, was the owner of Davidoff, and he brought me to Switzerland to play handball. Turn, he was financing my whole chemistry education at University of Basel. During that education, I met one of the most known chemists in the world, Professor Bernd Gies from Germany. And Bernd was the father of so-called radical reactions. His wife died on cancer, and after she died, he came to me. I was one of his best students. And he came to me and said, of course I'm sad that my wife died, but I'm very much desperate because with all my knowledge, at that time he was a president of US Science Academy, so one of the most known scientists, and he told me I was so desperate all the time as I was not able to support my wife to live just a bit longer, and decided to do a cancer research. We met together in 2000. I got unlimited financial resources, all this as a student at the time, and we started to work in a new area at that time. At that time, nobody knew what we actually started to explore. Today, the whole world knows what we did. We are the inventors and researchers who discovered how everything what lives, how does it grow. And we discovered that with the yeast first. So we discovered how our yeast is growing because of one specific enzyme, and we gave the name of that enzyme mTOR, target of rapamycin. But then we discovered that the same enzyme is present in human cells and in animal cells and many other cells, and we gave one mTOR, mammalian target of rapamycin. And then we started to think, is that enzyme also relevant for cancer? That was my PhD thesis, and the answer is yes. So the first molecule that we developed is a rapamycin natural product analog called today Afinitor or everolimus, commercialized on the market through the company Novartis. And it became registered in 2010 for several different cancers. It became a blockbuster drug, leading $4 billion every year. I got the offer to work in the management of oncology at Novartis, also other big pharma companies, but I actually wanted to be a chemistry professor, and I knew that we were just at the begin of a big thing. And many years later, it has been shown that I was right. So what did we do in the meantime? We discovered at University of Basel a twin brother enzyme of this guy mTOR, and this twin brother enzyme is called PI3K. In biochemistry, phosphatidyl-inositol 3 kinase. And this is a very bad guy. Each of us, we have that enzyme. The enzyme is actually in a very good shape now, not doing anything. But this guy controls whether you will get cancer or not. How? If there is a mutation of that enzyme in our body due to different reasons, this enzyme starts to produce a huge amount of sugar within your healthy cell, and because of that amount of sugar, the cell starts to grow uncontrolled, and it becomes a cancer cell. So sugar is healthy, we need energy, but uncontrolled production of sugar leads to boost of the energy, and that's a cancer cell later. So when we discovered that, we realized we have to target both enzymes, PI3K, to control the growth and the sugar production of the cancer cells. And I did that together with my sister. As I said before, we are all in the family as handball players and chemists. My sister the same. She came from Serbia as well to Basel to do the research with us together, and we discovered a new drug called bimiralisib. B, because we block both enzymes at the same time. Mira is the name of our mother. Unfortunately, she died on cancer 15 years ago. She had a very aggressive cancer, and we wanted that her name, Mira, is incorporated into the name of our invention. And we developed over the years this drug, and I'm very happy now to share with you the most recent news, 2 weeks old news. We are the guys now, first in the world, being able to cure a very aggressive skin cancer with a cream formulation. Wow. So, thank you. This is a phase 2 clinical trial, 46 patients. Patients at the early stage with this type of non-melanoma skin cancer. They have first the disease called actinic keratosis. I see most of you have type 2 skin. Most of you, except you actually, you have a better quality, so to say. But for example, I see you have a typical type 2 skin, which means if you get your skin exposed to the UV light, you have a much higher chance than me as well to get actinic keratosis, which is a precursor disease of non-melanoma skin cancer. If you don't treat that over the years, you have a high chance to get non-melanoma skin cancer. As most of the men, especially people with type 2 skin. The good news is, even if you get it, we can cure you with a cream. And that now activated a lot of pharma companies to talk to us, and we are now in process of getting the first multi-billion offers from different pharma companies. The idea is to sell that, to be able to develop a much bigger story called Swiss Rockets. When people ask me what is Swiss Rockets, are you competing with Elon Musk? We like to say with a joke, yes, we compete with Elon Musk, but just on a molecular level. So, shortly, Swiss Rockets is, we do rocket science with a rocket speed. We develop cancer drugs. This is not our first drug. We already developed several drugs, sold some of them as academic scientists to big pharma companies, like Affinito or Everolimus is the first drug coming from our laboratory. But in the meantime, we developed different cancer drugs, but also different vaccines. You will hear afterwards more about. So who we are as Swiss Rockets, we are incubator and accelerator. Incubator sounds nothing special, but what we do is a new business model how to perform as an incubator. So what we did, we partnered with different cantonal governments in Switzerland first. First partner was Basel government, then Basel country, then Bern, then Geneva, then Berlin later, Germany. We established a network of research partnerships, also with a network of local governments. We partnered with them, and we said, if we invest into the research at your university and make a blockbuster drug out of that, we will pay you some royalty payments, not more than 2%, but in return, we want your whole infrastructure for free. And we were able to succeed on that. So we are saving a lot of money not having our own laboratory infrastructure, getting access to the governmental infrastructure in Switzerland, Germany, and also now in the US. It's unusual to have that type of partnerships. Usually incubator get one place, they get support from the local government at reduced cost to use laboratories, and that's it. We have a much broader network, and we give our companies everything for free to use that infrastructure. They only need to pay a bit the office space, but the laboratory space, manufacturing space, we have access to factories which we can use for free, and in return we have to pay royalties when we succeed with some of the molecules that we develop. But what is also unique in that business model is that we accelerate. We are entrepreneurs, so we invest our own money in each of the projects. If I come to you and I ask you for a few millions investment, I would personally not feel well if I'm not investing minimum amount that I'm asking for. So we all invest in each of the projects. Together with other family offices, we invested so far 220 million Swiss francs to come to the level where we are today. And now we are planning to invest additional 300 millions. So this accelerator approach means funding those companies so that researchers can focus to the actually jobs where they are the best. They don't lose time traveling around the world looking for investors. We do the job, you focus to the drug development. And accelerator approach means as well, we hire the top executives worldwide, mostly from Roche and Novartis Basel. When they need few years to get retired, we identify them, we give them good offers, they come to us to be employees, and we give them good amounts of shares or options, normal salaries, not high salaries, but they get a lot of shares as they are financially independent. And then from the holding company Swiss Rockets, we outsource them to those subsidiary biotech companies which we are developing. And we control the operations. And that's what our investors like, because we literally control operations, and this is important, to be controlled by experts and not by financial investors or somebody who doesn't understand really drug development. So that's the concept of Swiss Rockets. When we do the clinical proof of concept, meaning phase 2, when we show the drug works, the drug is safe, then we go to the market, to big pharma companies, and we say, look what we have, are you willing to buy that? And in most of the cases, they buy it. If through some reasons we don't find the buyer, we do the IPO with those individual subsidiary companies, and we take 100, 200, or 300 millions, and we go to the market by ourselves, and then we sell the company later. That's the concept we do. So some people are asking me, are you VC fund? Are you a private equity growth fund? We are not. We call us entrepreneurs. We invest, we take the risks, we develop things, but you may say we are like a new venture of a private equity growth company. I don't like to use those words. We are just drug development entrepreneurs who understand also how to develop, especially a cancer drug. So what we do, we scout our technologies worldwide. We go to US, we go everywhere to Europe, we go to everywhere where we think there is a good research, good science. We send our so-called drug hunters. They screen everything, And when they recommend the projects to work on, then we have different committees, we approve them, and then we move with a rocket speed and a lot of power to move very quickly those projects. So that's how the process looks like, but I would quickly move forward. So what we want is, we know from this first drug, which I mentioned before, this drug was also able to cure certain cancer patients. We have today still patients alive thanks to that drug. But in many cases, we were able actually to prolong the life of those cancer patients, adults and children. But at the end, many of them died. It was a big success for Novartis, also for us as scientists to see the success of 4 billion revenues, to see how the drug helped many patients with those difficult to treat tumors. But I was exposed to many patients where I realized actually we have to change something urgently because many of patients are dying. So that's why we came to the new mechanism of action discovered by me and my sister. Can we block the production of sugar at the same time and the growth of the cancer cells? And we succeeded in doing that, and now we have a result in skin cancer, which is a breakthrough result. This is going to change the skin cancer treatment within the next 2 years. It's a phase 2, so we need to do the phase 3 with a higher number of patients, and then after that we will be able to globally register that drug. As a cream, also orally bioavailable drug is showing a lot of different efficacies across different indications, so we want to do that as well. But I will show you just quickly the overview of Swiss rockets. I don't need to explain you that there is a huge market for cancer. Every third person that you see outside is going to get cancer during its life. It doesn't mean that the person is going to die, but every third person will get the cancer. Many will die, and our goal is to prevent that. To prevent that and to have also new technologies how to treat those people which are going to get cancer. I will just show you one important slide, very simple slide, how we want to do that. So, when people get metastases, they are programmed to die, because this is a natural programming. Many of them will die. We realized with our technologies, if we exhaust the cancer cells by stopping the sugar production and their growth, we can help many cancer patients. We can even cure cancer patients, like in case of skin cancer. But when they really get metastases, and I didn't want to show slides how patients look like. If you see the PET CT scan, you see they have everywhere huge amount of cancer cells. They are black completely. They even get black skin because they are full of cancer cells. And we realized those patients, they have a very high probability to die. So we want to do something for those patients. And we came to an idea, what can we do? We came to an idea, okay, let's start to do something really new, really innovative. At that time, I already met my wife. Nadia is a nuclear pharmacist. She's one of the 5 people in Switzerland having license not to kill, but having license to produce radioisotopes for nuclear medicine purposes. So she was able at ETH Zurich to produce a new radioisotope as first in the world called terbium-161. And we realized if we put her radioisotope on our molecules that can go specifically to cancer cells, we realized in the first patients in Germany, we can eliminate huge amounts of tumor cells in those metastatic cancer patients. We showed that first in metastatic prostate cancer patients by men. And we said, wow, that's unbelievable. We published those data. It became a huge interest, that therapy. Today, this is the most attractive therapy in oncology, also in business development. Companies like our company, Torpedo Pharmaceuticals, they get multi-billion offers. So we showed now we can literally change the life of those patients. But then, we combined the knowledge with my wife, at that time girlfriend, so we combined the knowledge, we combined our private lives. We have a boy, small boy today, but we literally found out that the best approach to treat cancer patients so far, to our knowledge, is actually to exhaust the cancer cells by stopping the sugar production, and when they are so exhausted, to send the torpedo molecules. We call them torpedo because you inject them into a bean, they navigate literally like torpedoes until they recognize the receptors on the surface of cancer cells. When they attach that receptor, the molecular nuclear bomb is triggered to irradiate the cancer cell. And this works in humans, not only animals. It works in humans. It has been shown on many different patients. We showed that this works in ovarian animal models, in prostate animal models, in breast cancer animal models, but then we started to test it in humans, and we have realized this works in humans equally well. The beauty is, because they are targeted molecules, we call them targeted torpedo molecules, we almost do not have any side adverse events. For example, in prostate cancer patients, we only have a loss of saliva for this period during the treatment, because those receptors, they're also present in saliva cells. That's why we have this side effect. What I want to say, those molecules, they go super specific to the cancer cells. And if we exhaust the cancer cells and then we send the torpedoes, our killing effect super precisely is much higher. So we decided certain cancers to partner with big pharma companies. That's why we don't need a huge amount of money to put that by ourselves to the market. But certain cancers, like prostate cancer, we want to do by ourselves, because we literally believe that within the next 2 years, we will be also first in the world curing prostate cancer. First time in my life, I'm so happy to start using that word curing. You know, since 5 years ago, I was not able to say that. My goal was to say, yes, we can turn a deadly disease into a chronic disease. But now I am very free to say, yes, we can cure metastatic cancer patients. So the science is progressing rapidly. That's why we would also need to protect our data and everything exactly with good security systems. That's very important for biotech companies. And now I would also like to say where I see also the future in curative treatments of those cancer patients. So, as you know, today people are using chemotherapy, radiation therapy, surgical procedures, and also targeted cancer medicines with antibodies, with small molecules. But what is completely new is this so-called radioligand therapy. We call it torpedo molecules, radioligand therapies. And now we start to combine that, and we see there are breakthrough results, but I believe we will be able to cure a very high amount of cancer patients worldwide, statistically very high, if we even combine metabolic approach, meaning stopping the sugar production, radioligand approach, sending the torpedo molecules, and combining that with the immune therapy, different immune therapies, because the key is to activate your own immune system. We have seen in those human experiments, we have seen when people have huge tumors, even radioligand therapy it's not so strong to kill huge amount of tumors. But if you catch those tumors on time, and you destroy those tumors with a radiation power, with a energy supply power, like production of sugar, and you activate your own immune system, that's the best we have seen so far in all the experiments. And that's why we started to build a new ecosystem. That's actually the most important slide. You will understand with that slide our whole vision. So we have built Swiss Rockets as incubator and accelerator. We have built the company Tokyo that I have explained before, stopping the sugar supply, the energy supply, boosting of the cancer cells. We have built the Torpedo radioligand therapies. We made 2 factories, one in Denmark, one in Switzerland, to produce the radioisotopes for Torpedo. The name is Rocket Isotopes. But company RocketVax is very important for us. We started to think about cancer vaccines, utilizing new viruses, how to boost the immune system in cancer patients. That's what you're doing right now. And in future, I would like that every healthy person gets a vaccine. In future, the whole science goes towards that direction, that you go to your doctor and your doctor is giving you a high quality vaccine. I don't say that this is an mRNA vaccine. In my opinion, this is live attenuated virus vaccine, the traditional vaccine. And you are going to be protected for different cancers for the next 20 years. That's how I see the future preventing cancers. What we have done here, we started to validate our technology called reverse genetic engineering. And maybe you heard from the media about our product. We have a nasal COVID-19 vaccine. So when the lockdown in Switzerland started, with our technology in this company, we were super quick to make the recombinant SARS-CoV-2 virus. Recombinant means we artificially made a real SARS-CoV-2 virus. How did we do that? We took one of the very heavily sick patients from Italy at that time, took her blood, we isolated the virus, and then we were able to make from RNA of the isolated virus to make the copy DNA, and then the copy DNA we were able to synthesize, turn back into RNA. This RNA we injected into kidney monkey cells, and those cells produced the real SARS-CoV-2 virus. With that, we have validated our technology, and then the government started to ask us, can you also now make the vaccine? Yes, we can. What we did, this copy DNA, we just took the replication proteins and we made a castrated SARS-CoV-2 virus. We made a virus super safe because our vaccine can go into a cell, but it cannot go out. We castrated the SARS-CoV-2 virus, eliminating the possibility to replicate. And this became a super safe vaccine. And the US government, the Biden administration, decided to fund that project with up to $400 million as a next generation COVID-19 vaccine. COVID, although we are not talking about that, is still killing people. About 2,000 Americans are dying every week in the US. Because of the war and everything else, people are not talking about that anymore, but it is still a silent threat. Long COVID is also especially the problem. And now we would like to develop that COVID vaccine so quickly through the clinical trials to get in about 2.5 years the vaccine on the market. But it is just the validation of the technology because we can make also cancer vaccines. How? We can put the cancer proteins, specific proteins that only cancer cells have, we can re-engineer those proteins into the genome of a relevant virus. I give you our secret, what kind of virus we are using. It's a corona common cold virus. When you get once per year or 2 times per year super severe cold, this is mostly corona common cold virus, coming from the coronavirus family, but not dangerous virus. But what is interesting for that virus, it goes specifically to your dendritic cells, your part of the immune system. And those, they get the replication of the virus, and then they are exposing the virus proteins to your T cells, CD8 T killer cells. And those cells are killing the virus and the infected cells. So what we're doing, we're exposing also the cancer protein to immune cells. And when they catch that, they go to the tumor cells and they kill them. And it works in animals. Now we want to test that also in humans. But combining these 3 technologies, Manuel told me you have to show what really your vision is. That's my vision. And we are very close to show that. We already showed we can cure cancer here. We already showed we can cure cancer here. But we want to cure more than 50% of cancer patients. And how we do that, we combine the technologies that we have here. And before I talked with Julie about 23andMe, that's why I brought also this project. This is a US-based company. We are now organizing few hundred millions to buy that company, Complete Genomics, because this company, in comparison to 23andMe, looks only for they look for your origins, so they can tell you from where are you coming from. But if they look for certain genes regarding cancer or other diseases, they look for 2 genes. They look for BRCA1 mutation, for ovarian cancer and breast cancer, Angelina Jolie gene, and they look for Parkinson gene. What we are doing here, this company, they look for 30,000 genes. Did anybody do 23andMe test? Okay, you did it. How much did you pay? That's a good price. 150, that's a good price. But you get a very small amount of information.
I actually, because I did another one specifically for my athletic training and for my food, and I have to do another one.
Yes.
I can't remember if I can finish it off. You're right, it's quite basic.
Yes, this is quite basic, and that's okay price for that. But when cancer patients get their personalized medicine, they have to do, with the oncologist, they have to do the tumor profiling to see which medicine will react to that tumor cell. And you take the sample of the tumor after the biopsy of the patients, you give it to the lab, and they perform about 500 genes. And they say, okay, we catch this and this gene, here's the therapy. In most of the cases, this works. But you have to pay. In Switzerland, you are paying about 3,500 Swiss francs or 4,000 Swiss francs to do that. In other countries, 5,000. In Germany, it's a bit cheaper. In US, it's around 5,000. Remember, 500 genes. This company is doing 30,000 genes for $200. And in 2 years, it's going to be $10. So what we now want to do is to do the revolution in precision oncology, in precision medicine, because we can test, everybody can afford that. If your insurance says no, you can pay it. You can pay it. So that's the precision medicine. But visionary-wise, what do we want to do? I want that each of us gets this test. I just did it. I just did this test. And I see my origins. I was surprised that I have few Jewish genes, few Swiss genes, although I was always thinking I'm originally from Serbia. I have 54% of Slavic European genes, but the rest, I'm coming from Iran and Pakistan. I'm coming also some Jewish, this test can go to really true origins. And that's unique comparing to 23andMe. But it tests also all the possible mutations from 250 different cancer diseases that you could catch. And luckily, I was very happy. Now I know scientifically, I don't have any risk for any cancer at the moment. I also know that I didn't give any dangerous gene to my kids. I have 4 kids, so it is important to know something like that. And I can sleep now well. I catched with that test that I will get when I get aged, I will get macular eye degeneration, as many other people. But now I know that, so I can do certain things to improve my state. So what I want to say as a last information, so in a very near future, people will do that type of testing to prevent cancers. And we want to do that. But if we identify cancer patients that they didn't do that type of testing, they already have the cancer, we will be able to treat them in a new, innovative way with a big probability to cure them. But the real actually information is doing early stage testing, doing precision, targeted medicine. We almost do not have safety problems here. If you go for no melanoma skin cancer into Google, go to Google Images and put the word Aldara. This is the current product for that type of skin cancer, Aldara. Look at the face of those people. They're burned. They are totally burned. We don't have any side effect because we are specifically targeting the cancer proteins. My only side effect is dry skin during the 4 period of treatment, 4 weeks period. I have only dry skin there. So the science is moving rapidly forward. Targeted precision medicine is moving forward. Early stage diagnostic is moving forward. And when you get all of those data, then I would like also to do AI in a way to be able to predict and tell you, okay, in 10 years, you may get lung cancer. Check that once per year. If you get it, you get a personalized, live attenuated virus vaccine. You don't need to use those molecules, as you are still, you have only few cancer cells. We can catch them. You boost your immune system, and you're good. That's why I also like to start talking here about precision longevity. If people don't die on cancer, they will live longer. And that's why I call it precision longevity. So I just wanted today to give you a bit overview about the most innovative trends now in oncology and where the science is moving forward. And I can give you a good news because we show good results. Good news is science is progressing, medicine is progressing, but we have to work also how to improve in general the healthcare systems, how to improve the health insurances. That's the next goal. Thank you for your attention.
Very good. Outstanding.
Are you binding your isotope with your targeting protein?
How we are binding that?
Yes. I'm
Yeah,
just trying to
yeah,
imagine
it's
the
super.
steps behind it.
No, it's super simple. So first of all, you need to have a molecule that binds to a protein which is overexpressed only on a cancer cell.
Right. I understand how the targeting works with
Yes.
the protein,
But
actually
I
binding
understand.
with the
Yes,
isotope.
yes, I'm just trying to explain. So when you identify that molecule that goes specifically targeting that receptor, you also understand which part of your molecule goes to that receptor. So the part at the other end, you put, as we call it, linker. And to that linker, you put so-called chelate molecule. Chelate molecules, they can catch the certain elements, in our case radioisotopes, and the radioisotope is very, very much catched in that structure. It cannot go out of the molecule, and then you have a torpedo molecule. So simple inorganic chemistry.
So why the isotope and not chemotherapy?
Good question. Chemotherapy. My mom, I was treating her with our medical oncologist. She had pancreatic cancer. And we gave her first standard treatment. At that time, this was the gemcitabine, strong chemotherapy, in combination with another chemotherapy. That helped her to prolong her life. She was living for 70 months, which is unusual for that
But
disease.
you didn't have the targeting chemotherapy. It was just the general
Exactly,
chemo.
exactly. We gave her the second chemo, and we gave her the third chemo. The third chemo helped her, but 2 months after she died, because of the side effects.
For sure.
Another friend of mine, or my cousin, the same story. Third therapy started to work. They all died suddenly on the liver toxicity. My mom died actually on lung inflammation. She got a normal virus and she died on that.
Yes.
So chemotherapy doesn't differentiate cancer cell from a healthy cell. It kills also your immune cells. But what
100%, but you could do targeted
Yes,
chemotherapy.
you can do-
similar,
yes.
exactly similar concept
yes,
to this.
you call it antibody drug conjugates or peptide drug conjugates. People are doing that.
Yeah.
That's where Big Pharma is currently investing only into that concept
Sure.
and radioligand therapies.
Yeah.
But if you really compare that face to face,
Right.
we see better results with radioisotopes. Why? Much better results. We see 3 to 7 times higher potency. Because when the radioisotope comes into a proximity of a cancer cell, it destroys everything what is around 1 micrometer radius. It's a molecular, I call it molecular nuclear bomb.
Okay.
Chemotherapy, strong one cannot do that. So you literally destroy everything around 1 micrometer. That's why you have to have specific molecules coming to specifically to the cancer cells.
But then you are still damaging good tissue around the cancerous tissue.
As I said, no, because of that 1 micrometer. My only side effect for prostate cancer here, my patients sometimes they lose a bit saliva and they have dry mouth. That's my side effect. If I increase the radioisotope power from, let's say, 7 gigabecquerel to 15, I may have bone marrow toxicity, but I know that I should not do that, so I know my dose limiting ratio, and I'm good. I give it now only to that level, to the dose, dose maximum tolerated dose, and I don't have those side effects, and my drug is working.
So last,
Because you're weakening those cells with the sugar deprivation.
well, so that was going to be my follow-up question.
Okay, I'll tell you, but you guys should think very good. I'll tell you now more, yes.
So the sugar deprivation, though, should be enough.
I answered you while thinking about monotherapy. He came now with the sugar. That's my additional bomb. It's called a radiosensitizer. So I weak the cancer cells, and then they don't have any chance to escape.
So you can use a lower dose, even if it's next to regular cells.
That's our newest patent application. But that's the newest patent application we have. But we don't need to do that. We realized, in this case, 7 or 9 megabecquerel is good enough. So I don't need to lower it. But I have that as a backup mechanism.
Sorry,
But excellent questions, bravo.
sorry. Great presentation, but I have to say you're sleeping on the job too. You have to move your backside a little bit faster.
Yes.
Extraordinary things. I'm surprised that this is not more in the day-to-day information that we get, but that's all right. We're getting it now. Now that you can pretty much pinpoint exactly where we get cancer from, is there a way that, I mean, I can see that you're doing the tests and all that, but once you do that test, if you have it, even though you're identified early enough, we can do something about it. Is there a way, is there something that you can do to make sure that people don't do the things that create cancer in them? Can we prevent?
Yes. Are you a smoker? Absolutely. Okay. No. I tell you, my grandfather he was living 90 years long. He died because he went with a wet hair at -20, but he was super healthy, and he was smoking every day. So no chance to get lung cancer. When I have this information, then I have also probability, if I would smoke, I'm not smoking, but if I would do that, my chances to get lung cancer because of smoke are potentially lower. Why did he not get the cancer? Because he has so-called DNA repair enzymes protecting him. ATM, ATR, DNA PK, those are the enzymes which are, which can quickly repair the genome so that your cancer cell is not, that your healthy cell doesn't transform into a cancer cell. And I'm sure he had a very high concentration genetically of those enzymes. He probably had cancer cells, but it was repaired. Many people have that. That's why they are not dying of cancer. Many people do not have that, and they have much higher chance to get the cancer. My mom got pancreatic cancer. She was a heavy chain smoker. One of the key reasons to get pancreatic cancer is smoking. So in any way, I would encourage you to stop smoking because you will lower your risks. But with that test, in 24 hours, I can find out for $200 if you have those enzymes or not.
Tomorrow you're doing the test on everybody in this room.
We could.
The DNA repair enzymes. We'll tell you if you have it.
Yes. DNA repairing enzymes.
Okay, we have time for two more questions. Two more questions? Yeah, we have Paresh, yeah.
Hi there, I'm Paresh. I'm not a medic. Can you hear me?
Yes.
But I do a certain type of meditation which we know of. And in that meditation, to answer Maya's question, and probably not the audience or time right now to speak about it, I'd like to share at some point how natural therapy using meditation could assist in what you're trying to propose here, but particularly on why the cancer is caused. And that, if you understand that and you combine that to what you're doing, I think you've got a bulletproof system here. It's just extraordinary. Very impressive.
Thank you. Thank you. When I was a student, I wanted to learn yoga. I was professional handball player. All my friends in the class, they did yoga. So I said, okay, I want to learn yoga from a real yogi. I went to Trivandrum, Calcutta. And I spent... how? Kerala, sorry.
I come from Calcutta.
Kerala, yes, Kerala. I went there, I was 7 days there in ashram learning yoga. And I started to think in that direction. They do this, how you call it, when you sing every morning? How? Mantras, exactly, mantra. And they do that mantra every morning the same. And the frequency is the same. So what I believe is that certain frequency can kill the cancer cell. At that time, I was thinking about that. Today, this is not new. This is reality. Just recently, it was found that the Beethoven orchestra music can kill tumor cells very effectively, the sound frequency. So frequency is and that's what you are talking about. Exactly.
Because, well, particularly on the frequency side, so I'm talking about each chakra. Every chakra has a set number of petals, and those petals rotate a certain frequency. If you can harness or understand what that frequency is, you encourage the opening of those, because those chakras ultimately just manage those organs. And there's bundles of nerves which end somewhere in your body. So you can cure every single cancer just through meditation, should you care to. But we're not in a state where we can do that, so we have to have some preventative medicine while we encourage the
You cannot kill every single cancer. That would be illusion because cancer is much more complex. But as a prevention, would be very nice to have to meditate. Would be very nice to have a balance, because those frequencies, they literally do a positive impact in killing those cancer cells.
Okay, one last question for Vladimir. One last question or comment?
If some of you are investors, I can give you good news. In the past, we invested all the money here and here and here. People could invest only here, actually. We sold 30% of the company. One of the largest investors here is somebody you may know if you're from Switzerland, Urs Wittlisbach, Partners Group, co-founder. He bought 10% of that company. This company is the only one in history of Switzerland that got cantonal governments as private equity investors. It was a huge discussion in Switzerland, how the tax money was invested to buy shares of a company like this one. It's a progress for Switzerland. But we invested here and here, and recently we opened also all companies. Many investors, they wanted to invest only here or here or here to minimize the risks. So we opened recently all the companies, and only one product, this topical cream, for only one indication, skin cancer, with $10 billion market potential, is only one. I can tell you the drug works for at least 3 blockbuster indications for cream. We are getting now the first offers with multi-billion offer, but for friends and family who supported us the last 10 years, who invested also here a lot of money with our family offices, we decided to give them an attractive offer to invest here, or they can also invest into subsidiary companies, depending what do they want. Just that you have heard, as Manuel Ebner is co-founder, and he highly recommended to participate on that event, he also asked me if he could support some of them who are willing to be the part of the journey. The answer is yes. So you could, if you're willing, you could be part of the story.
So one last question just on rocket backs. There's a number of companies doing this with mRNA, and you're choosing not.
Yes.
Why?
If you Google CNN nasal vaccine, you will get the answers. We were in CNN explaining exactly that question. Why? Because with mRNA, you inject the mRNA, and this is producing the spike antibody in this case in large quantities, and you get antibodies, but less the T cell immunity. There is some T cell, but much more antibodies. And the lifetime of those antibodies is not longer than 6 months. But you have to inject it here, as we did it. Sure. What I am offering is a nasal spray vaccine. You do only this. You have the antibodies in the body. Here, mucosa. You have the T cells, and you have the T memory cells. So measles vaccine is actually activating the T memory cells. So my vaccine, as a nasal spray, will give you protection at least 2 years. So I have investors, they said, big pharma investors, your vaccine is so good that we will not make so much money, you know. But you will get the protection for at least 2 years, because you have activation at the entry side of the virus. And the virus, we have data, 3 Nature papers. That's why we got the money from the US government, up to 400 million, because we have the data showing that our vaccine works on a long term. The immune system is active here on a long term, and our vaccine can kill every single SARS-CoV-2 variant. mRNA, you have to upgrade it every 6 months to a new wild type mutation. You have to upgrade your mRNA vaccine. I don't need to do that. The disadvantage is I'm not so fast like mRNA yet, but I'm working on that to be equally fast very soon.
Listen, thank you, Vladimir. That's fantastic.
Thank you.